Okay, today we are going to talk about epithelial tissue. And you've probably seen this slide a couple times now. I talked about this on the very first day where we talked about the levels of organization. We talked about how atoms come together to form molecules. Molecules come together to form cells, and depending on the way that those molecules come together, you can form different types of cells. And then these cells come together to form tissues. As a reminder, there are 4 main tissue types within the body. There is epithelial tissue, connective tissue, muscle tissue, and neural tissue. And today we are going to focus on epithelial tissue. So what is epithelial tissue? And where is it? So it covers exposed surfaces, such as our skin. It lines internal passageways and chambers, such as the digestive tract and the respiratory tract. And it also has the ability to produce glandular secretions. So first, it's important to do a little bit of an introduction to cells and to go over cell theory. This slide is also here to remind you, that if you have forgotten your basic biology and what makes up a cell, what's inside cells, the different types of organelles and what they do, it would be a good time to review chapter 2 in your Martini textbook to get up to date on all of that. Because in this course, we do not review any of that. We assume that you know all about cells and what their organelles do. So there are 3 major components to the cell theory. The first one being that cells are the building blocks of all plants and animals. The second one is that all new cells come from division of pre-existing cells. So that could be through mitosis. And then the third component is that cells are the smallest unit that perform all vital physiological functions. And it's been debated in the past whether a virus would be considered the smallest unit. But in fact, a virus doesn't have all of the vital components. It actually needs many of the components that our cells have to be able to perform these physiological functions. So this time, cells are truly the smallest unit. Again, if you need to review any of these concepts, please visit chapter 2 in the Martini textbook. All right, so in addition to having cells in our body, most of our body is composed of fluid. All right, and so this slide is outlining the distribution of body fluid. Okay, so you can kind of see that here. So 1/3 of all of our body fluid is considered extracellular fluid. Extra meaning outside of the cell. Okay, so there is some fluid that resides in blood vessels and lymphatic vessels. This is a small portion of the fluid. But the rest of the fluid is considered interstitial fluid. Interstitial means between. So in between cells and among cells, there is fluid. And again, this makes up about 1/3 of the body fluid. Most of the body fluid is actually called intracellular fluid. This is within the cell. It is also called the cytoplasm. So 2/3 of our body fluid comes from within the cell. And as a reminder, the plasma membrane is what separates the intracellular fluid from the extracellular fluid. The plasma membrane is sometimes referred to as the plasma lemma. All right, so coming back to this slide again, it is going to be important to understand how cells come together to form these tissues. Okay, so how do we build tissue? All right, and so there has to be a way that cells can connect with each other to form these types of tissues. And there are several really important ways that cells connect to each other. So to make tissue, we need to connect cells to each other. And I have a few of these types of junctions on this slide, and each of them have a slightly different purpose. All right, so the first junction we're going to talk about are anchoring junctions. All right, so what anchoring junctions are they are going to connect one cell. So here's a cell over here, and here's another cell over here. They're going to connect two cells together. They're called anchoring junctions. So that's what you see in purple right here. These are the anchoring junctions because they look like little tiny boat anchors. That's how they got their name. And what they are doing is they are stitching two cells together, and they're doing so by holding on to the cytoskeleton, which is what you're seeing in reddish orange on each side of the cell. So it's holding the cytoskeleton and the membrane of one cell and the cytoskeleton of another cell and its membrane together. That's what these anchoring junctions are doing right here. These junctions are actually so tight and so strong that if you did try to pull these two cells apart, you would actually rip the plasma membrane. All right, we're going to cross these terms out here because I don't need you to know them. I just need you to understand what anchoring junctions are doing. There are two types of anchoring junctions. There are desmosomes and there are hemidesmosomes. Desmosomes are what we just talked about up here. They are holding two cells together. This is an electron micrograph of those anchoring junctions. So you can see the plasma membrane of one cell, the plasma membrane of another cell. You can begin to appreciate the cytoskeleton here, and then those anchoring junctions are in the middle here, and they are holding those two cells together. Another type of anchoring junction is called a hemidesmosome. That's what you're seeing here, or half. And what the hemidesmosome is doing is it's actually anchoring one cell, which you see here, to a basement membrane. So not only do we need to hold cells together to each other, but we also need to hold those cells down onto something so those cells don't just drift away. So that is what the hemidesmosome is doing. It's going to anchor a cell to a basement membrane, okay? There are two other types of junctions. So I'm going to go up here to the tight junction. The tight junction is going to stitch two cells together. It's going to do this in a way to control permeability. So there are instances where we need to be able to do that. I'm going to attempt to draw a blood vessel here. I'm going to attempt to draw two cells that are making up this blood vessel, okay? There's the blood vessel. And where you're going to find these tight junctions is in between those two cells there. Those two cells are going to be stitched together with tight junctions so that the fluid that is running through this vessel doesn't leak out. So that is how it's going to control permeability. The third type of junction is called a gap junction, and this is used for communication. So you can see these little channels here. So here is cell A, here is cell B, and you create these channels by these proteins called connexons, and as a whole, this whole unit is called a gap junction. This allows two cells to communicate with each other. They actually can share their cytoplasm as well. All right, so let's sort of define what epithelial tissue is. Epithelial tissue, it's commonly referred to as an epithelium. It is made up of one or more layers of cells that covers or lines the external and internal surfaces of the body. And I gave you a few examples already. You can find epithelial tissue on the skin. You can find it lining blood vessels. You can find it also in the digestive tract as well as the respiratory tract. Excuse me. And it consists of, we're going to eventually talk about the variously shaped cells and that they are closely arranged in either one or more layers. There are a couple of principal functions that epithelial tissue does. The first, again, I touched on this already, is to control permeability. And it does this through the actions of tight junctions. Excuse me. Epithelial tissue also plays a role in protection because it lines and covers surfaces to protect the underlying structures. I'll go on to show you that epithelial tissue can also form specialized secretions, and they do this by forming glands. And then lastly, epithelial tissue is associated with nervous tissue, so there's nerves that'll be present, and this is so that we can interact with the outside world, meaning we have sensation. Like if you think about our skin, we have sensation with the outside world, and this is necessary. There are key characteristics of epithelial tissue, and we can summarize these characteristics by using this amazing mnemonic called SPARKLE. Okay, it's an acronym where each letter of SPARKLE stands for a very important characteristic that epithelial tissue, all of them have. Okay, for the first one, which I've touched on already, but I'm going to show you another slide just to kind of drive it home, are these specialized connections. That's what the S in SPARKLE stands for. I'll go on to show you the P stands for polarity, the A stands for avascular. Remember, when there is an A in front of the word, it means without. So epithelial tissue is without vascularity, meaning there are no blood vessels present. Epithelial tissue has the ability to regenerate. Again, I'm going to go over all of these, but R in SPARKLE is regeneration. C is for cellularity, and L is for layers. So I'd like to touch on each of these again. Some of them you've heard, and some of them I haven't talked about yet. The specialized connections I've already talked about. We talked about these tight junctions. We talked about the anchoring junctions, right? And we talked about gap junctions. So you can sort of visualize all of these and where they might be present. These are those specialized attachments that are really important for epithelial tissue. The next letter is polarity. Okay, and I have a nice slide here. Oh, before we go there, I have this slide about gap junctions. This is to drive home the point that each of these were built by connexons. They are not individually called a gap junction, but the whole unit as in its entirety is the gap junction. These are those protein channels that are used for communication. This image is a freeze fracture image of two cells together. So if you freeze cells quickly and then you pull them apart, you're able to see these gap junctions a lot closer, so you can see these connexons. And again, as a unit, it is the gap junction. All right, now we get to go to polarity. All right, so what polarity is referring to is the idea that there is a top and a bottom to a cell. It is not just random, okay? So the top of the cell in this particular case is here, and it is referred to as the apical surface. This is the side of the cell that is either facing the surface, such as like on the skin, the side of the cell that is facing upward is the apical surface. It is also the side of the surface that is facing the lumen. What is a lumen? So the lumen is the inside of a vessel. So the lumen is here. So if this were a blood vessel, blood is flowing within the lumen of the vessel. So if you had cells all along here, okay, the apical domain is the side facing the lumen. That's that side. The apical domain is also the side where you're going to find microvilli if they're present and cilia if they're present. Remember those surface modifications that we talked about on day 1? Those are on the apical domain. The other domain is the basal domain. That is the bottom of the cell, if you will. And that is the side of the cell that is facing the basement membrane. It's attached to the basement membrane right here. So again, if we go back to the blood vessel, the basal domain is that side. It is attached to this basement membrane of that vessel. All right, and then I just want to come back up so that we're following the mnemonic. We talked about the specialized connections, and we talked about polarity. Now we are going to go through a couple other features with this slide, okay? So the A in sparkle was for avascular, which is what this is showing here. Epithelial tissue has no blood supply of its own. Here you can see the epithelium. That's what you're seeing here. These cells here, those are the epithelial cells. You can see the blood vessel here in the connective tissue. The blood vessel does not enter the epithelial cells. Therefore, it is avascular. As I mentioned, epithelial cells have the ability to have sensation. So they are what's called innervated. You can see the nerve here. Nerves are usually depicted in yellow. You can follow the nerve, and the nerve enters those epithelial cells. So this third point is that the epithelial tissue is usually supported by connective tissue. That's what you're seeing here, because they are not getting a direct blood supply from this vessel here, these cells are relying on diffusion of nutrients. You can't really see that when I'm writing in green. I should probably change colors there. Okay, so where are we? So we did specialized connections, we did polarity, we talked about it being avascular, a couple other features. Epithelial tissue has the ability to regenerate. What does that mean? So if cells are damaged or they die, this tissue has the ability to undergo mitosis and make new cells and regenerate itself. And this is not something that we should take for granted, because there are a lot of tissues in the body that have a very difficult time regenerating. Muscle cells, for instance, don't regenerate very easily. Neurons don't regenerate very easily. So this is a key feature of epithelial tissue. Another feature is what's called cellularity. So when you're looking at epithelial tissue, lots of times what you're seeing is cells upon cells upon cells. There are no fibers, there is no matrix, there is no ground substance. It looks like a brick wall. So it is made up of mostly cells. There's not a lot else there. Mostly cells. And then I'm going to go on to show you that epithelial tissue exists in layers. So there's either a single layer or there are multiple layers. Okay, so in addition to sparkle, and I touched on this when we were talking about polarity, there are two surface modifications that can happen on the apical domain of an epithelial cell, okay? And so I'm just going to write this here. So they are surface modifications that can happen on the apical domain. All right, so one of them is cilia, okay? So cilia is a structure that resides on the surface of the cell. It is a separate structure from the cell, And what it does is it exists as like it's a waving motion. So it has movement to it. So it's found in the respiratory tract. So the cilia will respond to, as you inhale air, there are lots of times dust particles there. And we need a way to remove those particles. And so what the cilia are doing is there is also mucus there. We'll talk about that more when we get to the respiratory system. But for right now, what you need to know is that the cilia are there. They are beating structures. Beating. Okay, they beat, and they all beat in the same direction, and they do so for movement purposes to remove the debris. So again, this is the apical domain. This is the basal domain. Okay. The second surface modification that you can find on an epithelial cell is called microvilli. These are actually part of the membrane. They're not a separate structure. So if we were just to draw it simply, this is what you would see. It's an extension of the surface membrane. And what that does, whenever you have a structure that looks like that, what you're creating is you are creating a greater surface area. So it increases the surface area of the surface of that cell. And this is important in certain locations, such as the digestive tract, because we are going to be absorbing a lot of nutrients. So we need a lot of surface area to do that. So these are two very different structures. They have very different functions, but they're found on the surface of epithelial cells. All right, this is another view of what microvilli look like. So under the microscope, it looks like they're kind of tightly packed in here. And if you did a scanning electron micrograph, you can really appreciate the greater surface area that is caused when you have these extensions from the membrane, right? You have so much more surface area than if it were just flat. This is what cilia look like when you look at it with higher magnification. So you can see these hair-like structures, right? They are going to beat. They are a separate structure from the membrane, and they're used for movement. All right, so let's start talking about how epithelial tissue gets developed. So all epithelial tissue begins as a simple single layer. So you see the single layer of epithelial tissue. And again, what would be the apical surface? Here. And what would be the basal surface? Here. And how do I know that? Because there is connective tissue here, and it is attaching it to the basement membrane. Okay, so everything starts out that way. The epithelial tissue all starts the same. And then, depending on the signaling, you can get various changes to occur to that epithelium. So, for instance, in this situation, because these cells reside in the respiratory tract, they are going to change their shape, which we'll talk about later, and their surface. They're going to add cilia there. In some situations, we want more than one cell layer. So here is an epithelium composed of many surface layers, many layers of epithelial cells. Okay, so when we have many epithelial cell layers like this can provide protection. So we can find this in the skin. So again, based on the signaling, we can change what's on the surface, we can change how many layers there are, and then finally, depending, of course, again, where we are, we may want to form a gland. And this happens by the epithelial cells. They kind of form an inward bud, and they start to form this gland that kind of dips down into the connective tissue. All right, so we can classify epithelial tissue by their cell shapes. And I'm going to go over 8 different types. So let's talk about, we'll go from simple to a little bit more challenging. All right, so there are 3 basic shapes that I'm showing you here. Squamous cells, they are very flat and kind of long, okay? We have cuboidal cells. They are shaped like a cube, equally tall, equally wide, and so forth. And then we have columnar cells. Columnar cells are named because they look like they're in columns, okay? That's what you're seeing here. And then when we have only a single layer, so if we have a single layer only, we refer to this as a simple epithelial, okay? So, for instance, if we were going to name the epithelium, which you have to get very comfortable doing, we would name this a simple squamous epithelia. If we were to name this, it would be a simple cuboidal epithelia. And again, simple columnar epithelia. So based on the cell shape, the cells can take on different jobs. And we will talk about that, right? Because it's going to be very important to have a specific shape depending on the area that you are in. Now, before we go into the specifics, I want to show you what it looks like when we have more than one layer of epithelial cells. And when we have more than one, we call it a stratified epithelia. So this is more than one layer. So your first job is just to look at it and decide if there's one layer or more than one layer, okay? And we're going to be talking about stratified squamous, stratified cuboidal, and stratified columnar, because in each and every one of these pictures, there's more than one cell layer. But let's look at this one first, okay? So you can see at the very top, there are squamous looking cells, right? I'm gonna change my marker here so you can see this. There are these flat cells right here. But as you move down, the cells look a little bit more cuboidal. So why are we calling that a stratified squamous epithelia? So what matters more than anything is what is on the top. You name an epithelial tissue by the cell that is on the top, not what is on the bottom. So this tissue is called stratified squamous. All right, now moving on to this, we have more than one layer, so it's immediately stratified. The top layer is cuboidal, right? You see that? So we would call this a stratified cuboidal epithelia. And then here we have more than one layer, so we call it stratified. And then we look at the topmost layer, and we see these columnar cells. So this would be called a stratified columnar epithelia. It doesn't matter that there are cuboidal cells down here, right? None of that matters. All that matters is what is on the very top. Please get used to knowing or being able to name the type of epithelial tissue that you're looking at. It is very important. Okay, so we've gone over 6 types so far. We've gone over them in cartoon image. I am, of course, going to show you what they really look like in a minute. Okay, actually, I do that first before I go over the other 2. I apologize. So what I'm going to do is systematically go through each and every one of these, okay? And the way I have set up my slides is I've taken this image straight out of the Martini textbook. And what these images do is it gives you locations where you can find the specific type of epithelium. It gives you functions, and then it shows you what it looks like in cartoon style along with the real histology. And so what I need you to do as you go through here and begin learning them is you always need to be able to name them or identify them on a practical. Okay, you will always need to be able to tell me a location. And in some situations, I will need you to tell me some of their important features or functions. And I will try to outline that as I go through here. All right, so the very first one we're going to look at is simple squamous epithelium. So that is a single layer of flattened cells. So I've listed off to the side a couple great locations that you can name for where you would find simple squamous epithelium. One is blood vessels. The other is in the alveoli of the lungs. Those are the air sacs that are deep in the lungs. That is where gas exchange occurs. Okay, so there's a really important function here. If we're going to be exchanging gases, right? If we're going to exchange gases, we would want a really, really thin cell to do that, right? Would we want a columnar cell? No, because that would take forever for those gases to exchange. So there's a nice structure-function relationship here. Okay, so name a location. One of the functions that a simple squamous epithelium has is that it can reduce friction, and it also helps to control vessel permeability. I really just outlined what the textbook said, but I put it here for you to see. Now, if you're looking at a simple squamous epithelium from the top down, it almost looks like fried eggs. You see that? The nucleus is in the middle, okay? That's what it looks like if you're looking at it top down. Here's a real histology image, same idea, okay? However, if you look at it in a cross section, you can then begin to appreciate how flat they are. So here is a vessel. You can see one of them here. You can see one of them here. Now remember, when you do a section, sometimes you're going to see them depending on the plane of the section. Sometimes you'll see the cells. Sometimes they won't be in that plane. So you can make out some of them. Okay, same situation here. You can begin to see those flattened cells because we are now looking at it as a cross section. So here are those flattened cells. Another great image is looking at the alveoli of the lungs. So these are the air sacs. This is where gas exchange occurs. So you can see this very, very thin epithelium. Again, this helps for gases to be exchanged really quickly. So a great location for simple squamous would be in the lungs. Okay. Then I go on to show you where you can find stratified squamous. So remember, stratified would mean there are multiple layers, more than one. And if it's stratified squamous, the squamous cell must be at the very top. And that is indeed what we see here in this picture. This is a stratified squamous epithelium. One location is in the tongue. Okay, so it's on the surface of the skin, the lining of the mouth, the throat, and the esophagus are some great areas to choose from. Because there are so many cells, it provides a great physical protection. Here is a real histology image. This is a histology image of skin. You can see all of those squamous cells stacked on top of each other. They do eventually turn into cuboidal, but it doesn't matter what's on the bottom. All that matters is the cell type that is at the top. All right, simple cuboidal. One cell layer thick. They are cuboidal in shape. So you can make them out really nicely here. Okay. You can find simple cuboidal epithelial tissue, epithelial cells within glands, ducts, kidney tubules. Any of those are perfect examples. Their functions, they're usually involved in secretion and absorption. You can find them here too. So this is a real histology image. Now, if I asked you where the apical domain is, since this is the lumen, right? The apical domain would be here, and the basal domain would be there. A couple other images, all showing the same thing, no new material. You can see these cuboidal cells, simple because they are one cell layer thick. Here is the lumen. That would mean that is the apical, and that is the basal. Okay, I think I've drilled that home, so I'm going to let up on that. Here's another great image. Okay, so you can see this is a tubule. This is the lumen on the inside. And you can see those simple cuboidal cells here. It's not exactly, I'm not drawing them as great, but you can appreciate that, okay? So locations are important. If it is listed, and then the function as well. All right, stratified cuboidal. This is rare. We don't actually have images on the microscope for you to look at, but I've provided images for you to see here. So you can find them sometimes in the lining of some ducts. Again, they are rare. Their functions, because they are line ducts, they are involved in secretion and absorption as well. You can kind of see the lumen. Here is the tubule. You can see that there is more than one cell layer here. So it is stratified. Okay, moving on to simple columnar. Simple means one layer. So here are those simple columnar cells. Okay, here are the locations. I highlighted a couple that I thought were easier to remember. It's really the gastrointestinal tract, or the digestive tract is fine to say, stomach, intestines, any of those are fine to say. Because these cells are really long, they are involved, or tall, they are involved in protection. Sorry. And you can make out that on the apical domain, there are microvilli. Here's a really great histology image showing you these simple columnar epithelial cells. Where would the apical domain be, and where would the basal domain be? Okay, here are a few locations. All right, another specialization that you can get in simple columnar epithelial cells sometimes, so you can see the cells here, right? Let me outline them. So those are the simple columnar epithelial cells. Occasionally, what you will find are something called goblet cells. And that's what you're seeing here, because they look like a little goblet glass. Okay? And what these cells do is they secrete mucus. So depending on where you are in the body, you may find lots and lots of these goblet cells. They are in the digestive tract, okay, because we need a lot of mucus secretion. So sometimes you will see that. All right, moving on to stratified columnar. Remember, stratified means multiple layers, and then the topmost layer would be the columnar cell. Okay, doesn't matter that there are cuboidal cells below it. These are also very rare. We do not have any slides in the lab with these. So you can see a nice cartoon image, and then you can see the real histology here. Again, make sure you can name a location. And because these cells are so tall and there are many layers, the role that these cells play are involved in protection. Okay, so we went over 6 different types of epithelial cell tissues, okay? And they were based on their shape and their layers, right? We did simple, cuboidal, squamous, columnar. We did stratified, squamous, cuboidal, columnar. There are 2 weirdos that don't fit in those categories. All right, so I'm going to show you those 2, and then we'll wrap up this talk. One of them is called pseudostratified ciliated columnar epithelium. All right, what does that mean? Okay, so pseudo means fake or false, right? So what we're looking at is it looks as if these cells are in multiple layers. But they are not. That's why it's called pseudostratified. It looks like multiple layers, but they are indeed in one layer. What's happening is that these cells are in various orientations. So they're not just straight up. Some of them are going this way, some of them are going that way, some of them are going this way. And so when you put the nuclei all there, it starts to look like there are multiple layers here. That's what this is trying to depict. But in fact, they are one layer. They're just in all different orientations. This is commonly referred to when we talk about the respiratory system. We'll talk a lot more about these cells. They are called PCCE cells. You cannot call them PCCE cells until you have written them out once so that I know that you know the entire name. You cannot leave out the ciliated portion of it if in fact they do have cilia. You would have to use the entire name. They are very common in the respiratory tract, in the nasal cavity, the trachea, and bronchi, okay? Their function is protection and secretion. Here is another histology image. Again, you can see, what side of the cell is that? It's apical. What side of the cell is this? That's what I mean by features. I just want you to know, sometimes it's easy to know, sometimes it's a little bit harder. So be able to, if you saw this image on a practical, be able to name it and tell me a location. And maybe I might ask you where the apical domain is. All right, the last kind of weirdo epithelium is called a transitional epithelium. You can only find this in a few places. The bladder and the ureters are a great place to name if I ask you about that, okay? And basically what's happening here is that it takes on a very different appearance depending on whether it is stretched or relaxed. So you can kind of imagine if the bladder was full, the epithelium might look like this, right? It's kind of stretched out. And when the bladder is empty, the cells kind of stack up on each other. Okay, so that's what they look like. They don't fit, it doesn't fit any of the categories that I mentioned at the beginning. Okay, so here, again, it's relaxed. When you start to stretch it, when the bladder becomes full, the cells start to kind of thin out a little bit. And you just have to be able to recognize what that looks like. I think I have one other histology image of what transitional epithelium looks like. So again, here they're looking like they're kind of stacked on each other because it's probably in its relaxed state. Okay, so I'm going to end this talk. Again, make sure you can identify all of the histology images, be able to name them, give me a location, maybe tell me some of the features if I've gone over them, and some of the functions if I may